Hello and welcome to this biology lesson on distribution of organisms. This is in AQA Syllabus B2, and in this video we will be learning about all these things listed here, particularly equipment such as quadrats and transects, how transect is used to estimate populations of organisms, as well as distribution of organisms. We'll also learn about how to estimate populations using this equipment, as well as we'll be using a lot of mathematical terms such as mean, median, mode and range. We will learn this during this video topic. All these terms we'll come to familiarize ourselves with, and we should be able to use it with ease to analyze data on organisms. Say, let's start with this. This is an example of to show what distribution actually means. And we look at an organism. In this case, it is daffodil plants. Now, if you imagine each of these flowers are daffodil plants, each of them represent daffodil plants. You can see they are not evenly spread in this habitat. Now, habitat is a place where living things live, and they get supported with all their life needs. So you can imagine that is the habitat for these daffodil plants. Now, distribution actually means describing how they are spread out in this habitat. If you look closely around the tree, you get very small number of daisy plants here. So they are thinly distributed. That's a very crude way of looking at it, but we look at numbers and figures later on. But for now, as long as you understand what that word means, that's enough. If you can see that they are very thinly distributed here, but as you go away from the tree, you get more population density. So they are much closely packed, and you can see the concentration and the distance between them getting shorter and shorter. So you get more daisy plants as you move further away from the tree, and that is an example of a distribution. We will then look at how to describe this with numbers, figures, and mean, mode and range, and those kind of mathematical terms. We can also estimate the population. So the first thing to understand and to be able to do is how to estimate a population in an area such as this. So next step is to be able to look at the steps involved in estimating population of daffodils. In this case, we are looking at estimating the population of daffodils in a habitat such as this lawn. Let's have a quick look at the list of factors that affect how organisms are distributed, that affect where organisms actually live. Now, for an organism to live, these factors are really important. Temperature of that habitat, it has to be comfortable and be able to the organism should be able to survive in that temperature. Now, some organisms do survive at slightly higher temperatures, but most organisms, or some organisms, can even survive in freezing conditions. But most organisms live in an average temperature. So it's really important factor. Availability of nutrients, food. Without food, no organisms can survive. So that is an important factor. So if you find a lot of organisms in a particular place, that could possibly explain it. But we need to look at carefully. That is the real factor. Amount of light, particularly important for plants. As you know, plants make its own food by photosynthesis using light, carbon dioxide and water. That brings us here. Availability of water, more important for plants, but animals, as you know, most of them depend on water. Either they drink it or they absorb it into their body, into their cells. So water is really important for most living things, including plants. Availability of oxygen, important for plants as well as animals. Plants also use oxygen for respiration. Animals use oxygen. Most living things, nearly all living things, use oxygen for respiration. Carbon dioxide, very important for plants, not that important for animals. but plant distribution can be affected by the availability of carbon dioxide. These are only the basic list. There are other things such as minerals and the predators. Say for example, if there are too many predators in a particular area, that can affect some living organisms. You will find less of them. Obviously, they get eaten in that place. You will not find a lot in that place. Okay, just to emphasize on that point, so not to overlook any other factor apart from these. So also look for other factors. These are the basic factors that are involved in determining how living things are distributed in a habitat. But you should also be in the lookout for other factors, other possible factors such as this. Okay, what is a quadrat? Quadrat is a square frame, usually made of plastic, and it looks like this. So there you see, here is a simple quadrat which has got even further divisions with wires inside. But normally it's placed randomly in a habitat, and then you can look at and count the number of organisms in this small area. So the idea is you use a lot of these quadrats, like 5 or 6 or sometimes even 10 in a habitat, and count how many organisms that are inside the quadrat, and use that to work out the average, and you will count all the number of organisms in all the quadrats that you've used, and use them to find the average in each quadrat, and use that number to estimate the population. Okay, you may not understand all this process for now, but all you need to know for now is a quadrat is a plastic frame of a specific area, usually 1 square meter. So 1 meter that way, 1 meter that way, and you multiply the area of this quadrat is 1 square meter. Okay, so how is it used? We'll see that later. So for now, don't worry about how the quadrat is used. We are actually going to look at this right now, how it is used. Okay, it's used to sample normally plants or organisms that move slowly. As you can imagine, you cannot use that kind of plastic frame to count the number of frogs living in a particular place, because the frogs will jump out of the quadrat and just escape and run away. Or you can't use it to sample something like a squirrel, and squirrels, before you even go near them, they just run away. So you need to use different techniques for them. Something like snail or plants are ideal, or even slow moving invertebrates such as woodlice are useful. You can use the quadrat to sample them, to live to find out how many of them live in a particular area. Let's have a look at an example of how to estimate population. So in this case, we'll look at the population of daffodils in this field, which is its habitat. Okay, so as I said earlier, we're going to be using quadrats to estimate, but we'll also be using some mathematical terms such as mean, number, but this is the same as average. So mean and average is the same thing. So we will be using these terms, so bear that in mind. Mean, which is the same as average, and we are going to be using quadrat to estimate the population of daffodils in this lawn. As you can see here, there are 4 quadrats being placed randomly. Now what randomly means is that there is no particular preference that you place these quadrats in. For example, you can't put them in a place where there are more, just because there are more daffodils and you should be able to get a better estimate. It just doesn't work like that. You have to generate a random place. Often it is thrown in a particular place with no preference, or sometimes use of something like a dice to select the area to sample. Very random places have to be sampled, and that is how these quadrats have been placed. So it's really important that you understand that these quadrats are placed at random. Okay, what that means is with no preference. Now let's look at how the sample can be estimated from just these 4 quadrat samples. So we have 4 samples, 1, 2, 3, 4. The total number of daffodils inside them are 1 plus 2, 3, 6 and 8. So the total number we have is 8 daisies inside. Total number 8 daisy plants, and we have 4 samples, so the sample number is 4 quadrats. Okay, alright, so what is the average number of daisy plant in each of the quadrat is the first thing that you need to work out. So the average number is, as I said, we have 8 in total. So the total number of daisy plants in all the 4 quadrats, 1, 2, 3, 4, is 8. But since we've used 4 quadrats, we want to find out how much per quadrat we have. So by dividing by 4, you get 2 per quadrat. Okay. Now, this quadrat is 1 square meter. So, area of 1 quadrat is 1 meter square or square meter. Okay. Now, if you know the area of this field, then we should be able to estimate the population. Let's say the area of this field is 70 square meter. Yeah, so we have area of the field 70 meters square. Okay, so now if you then multiply the average number by the total area of the field, you get the estimate of the population. So the population of, in this case, is daffodil plants equals the average per meter square or per quadrat times area of the field. Okay. Okay, so what do we get? The area of the field is 70 meters square, as I've already said. It's here. And we know the average per quadrat is 1 meter square. So what that equates to is 70 times 2, because that is the average in 1 quadrat. Okay, so that gives you an estimate of 140 daisy plants. Okay, I'm going to run through that again. We had total of 8 daisies in the 4 quadrats that we used. So the average per quadrat is 2, and the area of the field is 70 meters square. So what that means is you should be able to fit 70 of the quadrats in that field. And so the population is average per quadrat, which is 2, times the area of the field. So the area of the field in our case is 70. So 70 times 2 is 140. So we just estimated there should be 140 plants or the daffodil plants in this field. But if you count them, would you expect to get exactly 140? No, it's only an estimate. Okay, so we should not expect to get exactly 140. It may not be very accurate, it may be far off. It really depends on where we place the sample, and it is just an estimate. It's not the it's as close as possible to the real number. Okay, so how do we improve the accuracy? Accuracy means how close you are to the real value, and the only way to improve that is by taking more samples. Okay, so let's have a look at how we can get another estimate of the same number, the number of the population of daffodils here in the same field, but by using more sample. This time we have more quadrats. In fact, we have 1, 2, 3, 4, 5, 6, 7, 8 quadrats there. So obviously we have more samples. We should have a more accurate value of the population. We should have a more accurate estimate of the population of daffodils in this field. Okay, so follow the same procedure. We need to know the total number of daffodils here. 2, 4, 6, 7, 8, 9, 10, 12, 13. So we got 13 daffodils altogether as a total number in all the 8 quadrats. So total is 13. Okay, and so the average daffodil in each of the quadrant is 13 divided by, in this case it's 8 because we had 8 quadrats. Okay, so the average in this case becomes 1.625 per quadrant. Okay, so don't worry about this being a decimal number. You can't have 1.6 daffodil in each of the quadrant, but that's the way averages work. You do get these strange numbers. Okay, now we know the area of the field is or the total area of the lawn or the field is 70 meters square. Okay, so now for the estimated population, okay, so what would be the estimated population then? Is, as you know, the average per quadrant, which is 1.625 times the total area, which is 70, and that gives an estimate this time as, this time we have 113.7, so rounded up to the nearest whole number, because you can't have fractional daisy plants, daffodil plants, so you have 114 this time. Now remember, initially we had 140 plants, but this time you have 114. Now this would be a more accurate estimate without question, because we have simply used more samples in a second time around. Okay, just quick run through the calculations again. Totally we had 13 daffodils in all the 8 quadrats. So to find the average per quadrant, I have to divide by the 13 by the 8 quadrats, and I get this number, 1.625 daffodils per quadrant, which means per square centimeter. Okay, per meter square, or square meter. Okay, so we also need the total area of the lawn or the field, so we have 70 meters square. Now the estimated population is that average per quadrant times 70, which is the total area of the field, which means the field can fit in 70 of these quadrats, and you get a new estimate, which is 114, which is a lot different to the first estimate. Now we know this is more accurate simply because we used more samples this time. Often you will be asked to describe how you can estimate population using this technique, quadrats, using quadrats. Okay, so you should be able to describe in this like this. Okay, place a number of quadrats randomly, very important, around the habitat. Work out the average number of organisms per quadrat by dividing the total number of organisms by the number of quadrat. We have seen that in the previous example. Now finally, how do you estimate the population? Is multiply the average number of organisms by the total area of the field. Okay, so once you've done that, you get the area. Just a word of caution, not always you get the area of the quadrat as 1 square meter. Now, so I'm going to give you another example. This quadrat, in this example, the quadrat has got an area of 0.5, so half a meter square. So how would you find the estimate for the population of an organism? Now, average per quadrat is also given in the problem. Average area of the habitat is given as 80 meters square. So your first task is to work out how many of this quadrat will fit in the habitat area. So in this case, 80 divided by 0.5. So 80 divided by 0.5 gives you 160. So what that means is that is the scale factor. So the field is 160 times bigger than the area of the quadrat. So that is the number you have to multiply the average with. So in this case you will have the average number, which is here, average per quadrat, but this time you have to multiply by 160. So 2.5, which is the average number per quadrat, and the scale factor in this case is 160. So that gives you a total. So in this case you have 400 organisms. That's the total estimate of the population of that organism in this example. Okay, so not always you expect this area of the quadrat to be 1 meter square. It can be different. So your task is to find out how many quadrat will fit into the area first, and then you multiply by the average number per quadrat. So let's have a look at now the distribution. Let's have a look at a very simple example, the same field as we've been looking at so far. As you move away from the bottom of the tree, you get more densely populated daffodils. Okay, so the daffodils are more densely populated as you move further away from the tree. How do we explain it? So we've done the describing part. How do we explain it? As we've already seen, there are 4 main factors. The list of factors that we've seen earlier, I think it's more than 4. Now, out of that, which one is applicable here? Now, as you know, plants need sunlight, water, minerals from the soil, and carbon dioxide. These are the main factors which can affect distribution of plants. So where there are available more, you will find more of the plants. Under the tree, you'll get less sunlight. So that could be one factor. Now imagine sun is coming from an angle that can cast some shadow around the tree. So under the tree, if you go closer and closer to the tree, it's very unlikely that it's really shadow area where it doesn't get much sunlight. So that could be one reason. Less sunlight means less of plants such as daffodils under the tree, because the tree can block most of the sunlight. Okay, so that's another one reason. Secondly, the daffodils have to compete with the tree for water. So that's the second factor. So 2 things. One is less sunlight under the tree, and the second factor is less water under the tree. Very often you will have to do the 2 things. One is to describe the distribution. If it is data given, you have to describe what the data shows and then explain the reasons for the distribution. Okay, and the explanation comes from mainly from the list of factors that we saw at the start of this video. Here is that full list of the factors that we saw at the start of this video. Make sure you're familiar with all of these factors and be able to use it to explain the distribution. You should now be in a position to understand or state in very simple terms what that means, distribution of organisms. You should also be able to describe how to use the quadrat to estimate the population of organisms in a habitat. You know, the random sampling and estimating the population of living things in a habitat. Give appropriate explanations for the distributions you observe or what's given in the data. So if you're given a data, you should be able to explain to some extent and give possible reasons why organisms are distributed in the way they are in a habitat. Okay, and that is the end of part 1 of distribution. In part 2, we're going to look at more complex examples. So what you've seen so far is very simple examples and gives you an introduction into the lesson, into the topic of distribution of organisms. But we need to know, and we need to really come to understand and be able to do slightly harder and complex problems. So part 1 has finished here. I'll see you in part 2. Bye for now.